Good evening. Director of Expeditions, thank you very much. It's Director of the Academy, which is a department of TBA21 that we founded in 2011, and it's quite nice to be here tonight to give a little context and present the program. And usually when I present the program, I'm doing this alone, and I'm talking about art and science and conservation and policy making and all of this, and I'm alone. So tonight I'm really happy that I actually have a fantastic scientist with me, and then we will be followed by Peter Zinovieff and Andrew Spiro, his producer and collaborator, Andrew Spiro, with a fantastic sound piece. And so for the first time, we're actually, or not for the first time, but on one of very few occasions, we're bringing these worlds together. And they're both longtime collaborators of ours. Josef has been on 2 expeditions with us. And Peter and Andrew have been working with field recordings done in the South Atlantic, South Pacific, Chile. So yes, the TBA21 Academy brings together artists, scientists, conservationists, thinkers, cultural producers, and takes them into the field. And the field is, for now, the Pacific. So to take you there and give you a little bit of context and imagery, I'm going to show you a short film first, and then we're going to take it from there. Markus, sound? Still no? Markus, ratio? Yeah. It's Boris' computer. That's why. Boris, it's your fault. Beginning? Let's start from the beginning. Let's try again. Program to invite artists, scientists, environmentalists, thinkers, other cultural producers to come into the field. And the field is specific. TBA21 is a contemporary foundation that designs traditional categorizations in the artwork. And this is one of those projects. The current seeks to create culture outside of the traditional institutions and focuses on knowledge production rather than art production. By bringing makers from different disciplines out into the field, we collect a series of stories and point of views and create a kaleidoscope to unfold the place of people. We're making ourselves vulnerable, and we're going to show how the place is inspired and informed us. Water. Just the East Coast
in a nothing bag. You're going to ruin, you're going to mask, you're going to be thrown out, so you know nothing but our benefit.
I know that the ocean plays a very vital role in keeping the balance and even possibly reversing climate change.
We're at a point in time where we can say, okay, so it's like it's all over. We're at the end, and we're bombarded with terrible facts. But there has to be hope, and there has to be the possibility to do something about it. The information is there. It's just about how do we get people to care about our planet? How do we empower them to really take action and be an agent of change? That's the issue. These people here, they understand that the environment affects them deeply. And I think that there's a lot to learn from them in their closeness with the environment. How am I able to contribute positively to that? And I think that has to do with our awareness and understanding of the notion of nature, basically, and the environment.
In the right context, art is a very powerful weapon. And artists know how to use it to tell beautiful stories.
That's why we're putting artists who are magicians in communication together with scientists to really find a new method and find applicable solutions that no one in the lab would think of.
Traveling to very remote areas, immersing oneself in other cultures, and underneath the roof of a real oceanic experience is the only way we're going to be able to elaborate the stories and the urgency of certain emotions.
It's just an amazing place to be because you never know what's going to happen and you never know what's around the corner. It's worthwhile doing that. Push people to think outside of the box by taking them outside of the box. Artists has the freedom to imagine and reimagine and re-reimagine what is, what was, and what could be.
So this today is obviously not June 5 or 6. This was the last slide that we showed last week in New York during the first ever United Nations Ocean Conference, and that was actually at the end of the story. But okay. Anyhow, you all know TBA 21 is an art foundation with a program and a fantastic collection, and the collection is a large part of the collection is comprised of large scale, unconventional projects that are based on commissions. So in 2011, when we decided to found the TBA 21 Academy, we took these practices of commissioning, of producing, and exhibition making, and expanded on them. And we expanded on them first into an exploration program. We're bringing, as I said in the film, artists, scientists, environmentalists, other cultural producers out onto this boat to go on transdisciplinary, immersive explorations. And the idea is to engage with places and communities under certain curatorial aspects. But the underlying theme of this program is always how to recommunicate the effects of climate change on the ocean and its inhabitants and the people that inhabit its shores in a more engaging, inspiring, and empowering way. And we firmly believe in the power of exchange between disciplines. And I firmly believe in the power of the arts to be a connector between these disciplines. Artists, and artistic research can traverse these disciplines and dive into them and connect them on a larger scale, that these disciplines, through the trajectory that science has, can never do. And these are the conversations that we're having on the boat. We've tried to stay away for a while from exhibition making. So this exhibition that you see here is the result of 6 years of exploration, of research, and now of commissioning. It takes different vantage points to look into and elaborate the different topics around these commissions, around the works, but they all look at the ocean. Why is the ocean important as a topic, and why do we as an art organization look at the ocean? The ocean covers 72% of this planet, and every other breath that we take comes from the field that scientists very happily say unknown unknowns. So it's a very wide field. It's also obviously it's an element that is very uncommon to us and unfamiliar to us. It's something that is permanently in flux and in permanent movement, which is something that is hard to grapple with for us theoretically. And so this makes it a perfect exploration ground for a transdisciplinary experimental program like we're running. Josef, you've been with us twice. First time you came with us to the Galapagos. Second time you were with us in the Marquesas. They're both very remote places. The Marquesas supposedly the most remote island group in the world. And I'm already getting turned off. I'm obviously running over time. But one of these topics that is always coming up for us when we meet each other is collaboration between species, cohabitation of a space, which could be our body, and obviously this is also something that is that may be a method of adaptation to climate change, to use biomimicry, or your field, genetics, to counter or to cope with climate change. Tell me, how do you have a very interesting point on or view on collaboration and cohabitation? Why don't you tell me a little bit about the bacteria in me?
Oh, bacteria. Oh, okay. Such a nice day, so let's just exchange bacteria, shaking hands or something like this. So the bottom line is we are of course not an enclosed organism, or how did Leibniz call it, a menade? We are not, but we actually play ping pong with evolution, and so as we sit here, we probably exist. We have around trillions of cells. So we have around 100 billion brain cells and 200 billion astrocytes, which are different types of brain cells. And every day cells are born, millions are dying, but despite all of this unbelievable complexity, there are around 100 times more bacteria live with us than our total cell number. And bacteria are real chemists, so they do chemistry we cannot even dream about, and around 20% of our chemicals in your blood, as you sit here, is actually coming from bacteria. So somebody actually mentioned, we are actually not the end of evolution. The bacteria are much more clever than us. So they use us to run around, have a career, eat for them, and they might have even invented sex so they can go on holidays sometimes. No.
We're still an art organization, so this is up to your fantasy, right? Well, if the bacteria runs our bodies, right, then it is this. I mean, coming back to the United Nations, no, where we were last week. It's the question is, why do we and who does all these policies and the legislation? So we just to bring it back to the program for a second is we went to the United Nations last week. It was the first ever ocean conference there, and we brought our practice like a bacteria into the into the organism of the United Nations. And it is quite stunning that if you go there, and it doesn't matter if it's a non-governmental organization or if it's actually a legislative body, the communication, the terminology that they use is extremely technical, right? So now we've all agreed on blue economy, and we've agreed upon sustainable fishing, and we've agreed upon many of these fancy words that are used, and everybody thinks that by using these words, the something is going to change, but it's usually not, right? So you sit in the United Nations and you sit within these meetings, and everybody drinks out of plastic cups, and everybody eats food that is wrapped in plastic, but they discuss plastic pollution. So it's my understanding that it is super important to bring a different form of language and a different form of visual language also to these bodies. That's why we are applying ourselves to the United Nations. And the introduction and the introduction of spatial dimensions, of aesthetic dimensions, but also the reintroduction of let's say a political dimension of civil society is something that is super important. For you, coming from the scientific sector and having a clear vision on what we understand is reality today, right? And then you see the distance to policy making and politics. How do you bridge this gap also as a let's say a publicly funded institute?
So yeah, so actually what you guys are doing is spectacular, and I think it's very, very important. So I used to be many years the only Austrian being a young global leader with the World Economic Forum. So I was every year in Davos. I met all these guys. We actually saw the first screening of Al Gore's famous movie now. So before it was actually released. So we always wanted to save the planet. You know, the whole thing was how to make electric cars, for instance, sexy. So, you know, how to get laid in a Prius, basically. So how do you do this? How do you market? How do you bring science back to, you know, to the public, where it makes, where people get intrigued by it and, you know, might start buying a Prius. So, but on the other hand, it was lots of bullshit talk. I mean, you know, this, at the end, you know, it's, you know, we come all of this chemical scum which surrounded this weird planet we call Earth now. And somehow we as humans have developed some intelligence and creativity. But at the end, of course, we are responsible for the entire planet. And in that sense, you know, it's nice to drink coffee and have fancy words for certain things. But, you know, I think we should all feel responsible and actually do something. And, you know, to have this particular interest, you know, one nation has this interest and the other one this. What do you do in you know, saving the oceans, you know, or climate change? You know, there's not the Austrian climate or German climate or US climate. It's all over the planet. So I think I'm really putting my, you know, my hat off from what you're trying to actually change that. You know, go to United Nations and actually change this. So good luck to you. I miserably failed many years ago, and so I do my science again.
So was that a whole bunch of scientists sitting together thinking about how to get laid in a Prius?
Yes.
It was scientists. Okay, good. Wow. Okay.
No, it was actually mostly, this really came from, you know, people who are running CNN and some of the big networks. Because it's one issue to have scientific fact. The other thing is, how do you actually transport science to the public? And of course, you can transport science to the public with art, what you're doing, which is brilliant, or with other ways, you know, just marketing slogans.
Well, I think to bring real change, no, we have to radically change our perspective, and then also our action. And art has, through the centuries, been a vehicle to change our perspective, literally from flat to depth and others. But we strongly believe in the power of art as a vehicle of communication, but also to drive action. And that's why we're making this combination between artists and scientists to find out really what is going on, apply this to the field, and then work with it in an unconventional and unforeseeable, hopefully inspiring but empowering way. I saw that you have a slide in your presentation about the tree of knowledge. We've, as I said before, or I've said in the film, this program started out as a knowledge production, but when you take artists into the field and you work with artists, at some point they will create a work. So after 6 years, I said after 6 years of running the program, we finally have an exhibition. And if you haven't seen it, I would invite you to go and see it. 7 new commissions and 5 positions from the collection. Josef, I'm gonna change this screen for you, and there you go, the tree of life and knowledge.
Oops. Yeah. Anyway, so I think can I walk around, or because if I sit, I don't know, I probably explode. So let's. So good. So you have to suffer, and now it isn't. So I hope I don't bore you completely, because this is my life. What we do since many years, how we actually think we might revolutionize evolution. So I will tell you what we're doing to do evolution in an afternoon. So basically do millions of years what nature is doing, so what we can actually do now in 2 hours, basically. And they're always inspired by one of my favorite movie directors, Peter Greenaway, made this amazing movie, The Belly of an Architect. So when he asks him, what's important in life, anyway, I do believe some of the things we do are important, so don't kill me. So for me, of course, living in Vienna, this is one of the iconic paintings, and also because of the grand Jewish tradition of Vienna. And in the Jewish tradition, we have been driven out of paradise, so we have no eternal life. Eventually, we need to die, even if we don't want it and like it. But that's what nature is doing to us. But through fundamental understanding how the world works, in science, in arts, to figure out these little things and the grand things, we might be able one day to gain some years of life back again. Some years of good life, of healthy lives, with a grand vision, at least in the field I am. I'm a genetic engineer. So in my engineering business, my grand vision is that we all live to 90 or 100 and still play soccer, and we still score a goal. And I still remember where I'm home afterwards. So that's basically the grand vision. And it's very important for our society, because in the 20th century, as a population, we gained around 30 years of life. So evolution has never made us to grow older. But now we have the society with lots of people who go older, and now we have all these issues with it, with dementia, with Alzheimer's, with neurodegeneration, with diabetes, and so on. So if we cannot figure this out as society, probably blow ourselves completely out of water. And the other reason why I like this iconic paintings of Klimt is this one. So Klimt, and you probably know this much more than I do, was heavily influenced by scientists. So in 1880, this place where we sit here in Vienna, Vienna was the center of the universe of biomedical research. So this was the place. So basically the Harvard for medicine was here. And the reason was because there were some people who completely revolutionized how science was done. And one of the guys who did this was a guy called von Rokitansky. And von Rokitansky was a pathologist. And von Rokitansky did in his lifetime 60,000 autopsies. And by doing so, he was actually the first person ever who could say, you know, the heart does not sound well. And if actually people died, they opened up the dead body and then could correlate it with some disease. So this totally revolutionized how we actually understood disease and the human body. And one of the students of Rokitansky was a guy called Zuckerkandl. And Mrs. Zuckerkandl ran the big art salon in Vienna. This is where Klimt used to go. And so Klimt got to know Mr. Zuckerkandl. And Zuckerkandl was an embryologist. So he let Klimt look in the microscopes. And what Klimt actually started to paint is eggs and early embryos as decorative art for the women. So if you actually look at the dress of this woman, this is an egg which undergoes meiosis, as first cell division, because he actually saw it. So all the guys, always sperms, and all the women, decorative art is always early embryos and eggs, inspired by science. So basically, what is now considered, one of the grand decorative artists, was actually because he looked through a microscope and saw little eggs growing there. So another artist, and this is a great book if you want to read it, of Eric Kandel, who is on the head of my advisory board, so he's actually helping us to get back to good science in this country. And this is another classic painting of Kokoschka, and it's very famous because when he painted it, the family sent it back to Kokoschka and said they don't want to take the painting because the guy, he painted the guy and he is not real. And the reason is if you look at the hand, so the hand is hanging down and also the one eyelid is hanging down. So Kokoschka painted him having a stroke. But the guy actually didn't have a stroke. A half year later, he had exactly that stroke. So the artist actually saw already what might happen in the future. So, and I also have to say to, to Markus and Francesca, I'm a little, biologist and scientist, so I would have never had the chance to see some of these most spectacular places in the world. So this was our Marquesas expedition. So you can see actually the boat broke down, and so we had to bring them all back here. So this is where we climbed some mountains. So these are the people on the expedition, and this painting I like. So this image I like. So it's actually Francesca in the middle of the the jungle in one of the remote islands. There was this sign of a chocolate maker from Eastern Germany, and so he had actually made chocolates. No, are you sure? And some years ago, I had the pleasure to actually go to Galapagos, which, of course, for me as a biologist, is like the mystical place of biology, where Darwin, of course, among other ideas, developed the idea of evolution. And this is a cormorant which undergoes reverse evolution. So these guys actually behave like penguins now. So they cannot fly anymore, and you can see the wings are becoming smaller. And I also mentioned this because 2 weeks ago, actually, the genetic mutation was sequenced, which is the cause why these guys actually undergo reverse evolution. So it's a gene which regulates wing development in chicken. So, and what we want to do is learn from evolution. Basically, I think good science is not the idea how we believe the world works, but actually to observe nature. And of course, nature had millions of years to play with genes and play with adaptation, play with models of body plans and morphology and metabolism, and basically come up with solutions to life. So this is basically what we want to learn from nature and learn from evolution to fundamentally understand how this world works, how we work, and how we gain some life years back. And you know, during discussion, I already told you we exist of trillions of cells. So how do we do this? How do we actually figure out how this works? You know, every day 10 millions of cells die on me, 10 millions of cells are born. So if you have to imagine a machine where every day you have to exchange 10 million pieces and it works for 80 years. So it's you know, totally spectacular how biology works. And this is a single cell. So the complexity in a single cell. So how do we ever figure this out? A single cell, there are trillions of them, they all work together to make this body which stands in front of you, which allows me to get up in the morning, you know, do my things, be half intelligent or not. So how do we ever figure this out? So, but fundamentally, every life on this planet is born out of genetic sequences. So there are 4 letters of life, and the sequence of these letters basically make a human or a mouse or a plant or a virus or a bacteria. So based on this, these 4 letter codes of the universal life on this planet, so the idea is, of course, and of course it's more complex, that if we figure out the function of the genes, which code for a human, or why we look as we look, or which code for a mouse, how they look. So maybe we can figure out how it works. And again, there's this around 3 billion letters of life. So this 3 billion letters which make up the human genome. So recently I was in Cambridge in the UK. There's this beautiful museum on the history of genetic sequencing. So in the year 1990, we had technologies that would have taken 2,000 years to read these 3 billion letters. So now there are sequencing machines which look like an iPhone, which can sequence you in an hour. So within 25 years, the technology has exploded that we could not even read reasonably human being to basically reading in essence, and it's happening very fast, in essence reading every organism on this planet, from plants to bacteria to different humans and thousands of people who have cancer, to figure out the genetics of evolution, to figure out the genetics, for instance, why birds start to sing, which genes turn on singing behavior, and so on. And the other little thing is, of course, living on this little planet Earth. So if you would take me through the, you know, grind me up and take all the DNA which comes out on the bottom and then tie it together, so you would actually get 10,000 times to the moon and back. So this is the length of the DNA which stands in front of you. And the other thing, you would actually find that maybe only 5% of the DNA which stands in front of you is actually human. The rest are bacteria and viruses and fungi which live with us. So I don't think we're the highlight of evolution, but you know, unlike Douglas Adams, who believes mice are running us, so it's maybe actually bacteria who are much more clever. And you have to actually imagine what happens in our guts. So our guts are basically a whole planet of bacteria. So there are trillions of different bacteria in our intestines, around 4,000 different species, and they live in little towns and little villages. They help each other. They live in symbiosis. They're actually the good guy. Many of our vitamins we need are made because of our bacteria. So at the end of the day, we would be seriously screwed if we wouldn't live with this bacteria in our intestines. So, and I don't bore you with this little thing. So what we can do now is, I'm a genetic engineer, so we play on the piano of genetics. So one of the organisms we like are mosquitoes, this little thing. And no worry, we are very good genetic engineers, but we still have not managed to make flies with teeth. So this is Photoshop. Luckily, one day we make them, so then beware. If we sit out here, and we release flies with teeth, but it's not there yet. But the grand vision, of course, is why we use this, is because around 70% of our genes we can find in flies again. Evolution. Because we come from this evolutionary tree. So we can make flies sick, so they can get fat, they have heart failure, they learn, they can memorize things. And the idea, of course, is that fundamental mechanisms, how a muscle contracts, how a heart works, how nerve cells talk to each other, have to be conserved in evolution. That's why we can actually use these animals to figure out fundamental mechanisms, how we work. So this should be a heart. So it's a fly heart which beats 80 times a minute. And doing this and shutting down every gene, we come up with a subway map of London. So this was a 5-year experiment. 2 million flies analyzed, the entire genome mined of the fly. So this is the first genetic map of every gene which controls how hearts function. So, and I think I'll leave this with, so evolution on a lazy afternoon. So when we did all of these things, we learn from nature. We make mutant mice. So this is where I come from, genetic engineering and stem cells and animals. So we made some of the first mutant animals in 1992 when I was in America. So we play on this piano for genetic engineering for a long time. So we had no idea, for instance, that Dolly was possible. So there were some interesting developments over the last year, cloning, human cloning, of course. And one day, one of my students said, so why don't we, instead of learning from organisms in evolution, like the fly or the mouse and other model organisms like fish, for instance, fish are really cool. Goldfish, you can cut half of the brain off and 2-3 weeks later they grow a brain again. So many organisms can regenerate the tissues. The famous axolotl, you can cut off a limb, 2-3 weeks later they grow an entire tissue again, with nerves, with bones, the whole thing. So I think we can actually learn a lot. So one day, we regrow our brains again, hopefully. But then my student came to me and said, why wait so long? Maybe we can develop systems where we can play evolution or do evolution in an afternoon. So mine the entire genome just, in the laboratory very fast. And this was, of course, a totally crazy idea, but we did it anyway. And the reason was this. So yeast. So when I drank a beer, so the beer brewer is actually the first biomolecular engineers. So Saccharomyces cerevisiae, yeast, every biochemist and people who do genetics they have been using actually comes from Carlsberg because they wanted to make a yeast which is stable so they can brew beer. And out of the stable yeast, the geneticists actually took it and played with the yeast genetics. And the reason why yeast became so interesting is this. At a certain life cycle, they have a singular genome. So if you have a mutation in this genome, then it's kaput, right? So as we sit here, we have 2 genomes, 2 copies, which we call diploid, D double, one from our father and one from our mama, right? So when a sperm meets the egg, we get the genome from our mother and the genome from our father, and if one gene is kaput, the other one is in a reasonable outbred population, okay, and this is what keeps a population healthy. And so I now make a statement as biologist: nature wants diversity. Nature needs diversity. Nature needs outbreeding. So the more people we actually let in our country and let them breed with us, the better we are off in the future. So this is what we need. So populations which are inbred, always the same, eventually we run into genetic bottlenecks and then we are in trouble. But this for us as a geneticist is a problem, because we need to change one gene, one copy, and then the other one, and this takes forever. For instance, if you make a mutant mouse, it takes around 2 years to make this, with all the engineering we do with homologous recombination and stem cell engineering and blastocyst injections and chimera formations. I won't bore you with all the details what we're doing, but it takes for a long time. So the idea was, wouldn't it be brilliant to make a normal mammalian, as we are here, we are mammals, a mammalian cell which has a singular genome? Because if we would have this, then we could make mutations, and in one afternoon we could mutagenize the entire genome. And actually what evolution has been doing over tens of millions of years, we do in the afternoon in the lab. So we went for it. So again, back to the eggs, because eggs have a singular genome, right? Before the sperm comes, and when they get combined, they have 2 genomes. And so we thought, why not trick the eggs to believe there was actually a sperm? We have this little tricks how we do this. We actually give them 5% alcohol for 10 minutes, so it's not a joke. So you can actually take an egg which has a singular genome being ready to be fertilized. We give it 10 alcohol, it turns on a kinase called PKC on calcium flux, so I can explain it at the molecular level. And the egg, believe it has been fertilized, starts to grow a little embryo, but has never seen a daddy. And out of these little embryos, we reclone a stem cell. Stem cells are these primordial cells out of which everything can develop. So to make the story short, so we actually broke a paradigm of biology. So we grew a cell which has a singular genome, which is a perfect stem cell in mouse. Recently, actually, in the journal Nature, a group from Israel and New York published the same is possible in humans, the same is possible in multiple species. So based on this little technology we developed, it's very easy now to develop perfect stem cell with a singular genome. And now we can really play evolution in a day, because we don't have to worry about the second copies, we mutate. I hope I don't scare you completely, but I show you why we're actually doing this. So what can we do with this? So what we do with this is we take, and we do this all the time now, so we take 1 billion of the cells with a singular genome. And then we developed some systems with jumping genes, or some of them are called actually sleeping beauty, very interesting names, or HIV-based viruses which jump into the genome, or other retroviruses we use. And we basically can make 50 million mutations immediately. So as we sit here, we have around 25,000 genes. So we do this, that each cell out of the 1 billion has a single mutation. So you can just do the mathematics, 50 million mutations in an afternoon, where we actually only have to mutate 25,000 genes. We trash the entire genome in a day. So it's possible to do this. And we actually played some little games. So it's like a genetic barcode. It's like going to the supermarket. You can actually read it out again, the genetics. We do 4D Sudoku sequencing for aficionados of you. And then we go to the sequencing machine and figure out how this works. So I show you an example. Artemisinin, the most widely prescribed drug in the world for malaria. So it's the best medicine against malaria. Nobody really knows exactly how it works. It's also very toxic. And this woman, Yu won 2 years ago the Nobel Prize for finding artemisinin. And it's also actually really interesting little story, because in the 60s in China, Mao Zedong wanted to wipe out malaria in China, which was very prevalent. So they put a lot of the scientists, from the geneticists to the chemists, to the traditional Chinese medical doctors, to figure out something which would help against malaria. So Yu was a traditional Chinese medical doctor who read a text which was 1700 years old. And in the text, 1700 years old text, it had a line: if you take this particular plant, cook it in a particular way, you get the substance which helps against high fever, which looked like malaria. So she did it, she made the chemical out of this, and this became actually the best medicine against malaria on the planet as we know it. So reading sometimes old history. So there's lots of value. And the reason also why I mentioned this is, so there's a lot of value in plants, in nature, in going to the Marquesas, exploring plants. This has medicinal function and so on. So there's a lot of here too, yeah. So again, this is how it works. We take our cells, give them artemisinin, it kills because it's actually toxic. Nobody tells you. The Chinese use it for cancer therapy. We take the cells, mutate them, give them artemisinin. Some cells survive because now we have mutated genes which make proteins which are essential for artemisinin to kill. So we go to the sequencing machine and we found a single pathway. And actually finding this, we believe we can now explain why it works in malaria. And we can actually explain why it works here and might be able to explain the new resistances which develop in Thailand, in other countries, because we have a huge problem on the planet if the resistance against artemisinin for malaria, because there's no other drug which really works at the moment. So that's why we do this, to figure out how the drugs work, to figure out why there might be resistances. And if we know this, maybe we can make even better drugs. And we do this now for other little viruses I love. For instance, Zika virus. So when we were in French Polynesia, I talked to the guys. So actually I asked them what they do against Zika. They said they do nothing because everybody got infected already there. So 2-3 years before the virus got to the Americas. Of course, it causes, if women are pregnant, brain defects in the babies. So we work now on Ebola virus. So no worry, I don't have it in my pocket. So this is, it's only in my lab. So if you want to see Ebola, so this was a bad joke. So we work on Marburg viruses, which is actually even worse than Ebola, and many other really cool things, like rhinovirus, common cold. There's no drug against the common cold. It's the most common disease on the planet. And nobody knows what to do against it, but just, lie in bed and just sit it out. So again, the whole idea is, with this system where we can play evolution in a day, we give our cells, for instance, the Zika virus, it kills them, we mutate, cells survive, we go to the sequencing machine, 3 weeks later we know how it works. So we even have new technologies where we cannot just tell that's the protein which is responsible for Zika to kill. We actually can tell you the amino acid responsible. So we can now go to single amino acid revolution and resolution how these viruses work. So it's possible to find the molecular haystack in the grand ocean of life with these little things and shortening the time of that we study evolution. So the other thing we actually do is, and maybe I skipped this fast, is what can we do with humans now? So can we translate some of this genetic engineering we are doing nowadays to humans? And one thing which happened so there are three large revolutions which happened in science, in life sciences. So one was we learned how to read the genomes of species, of worms, of humans, of bacteria, of mice. So there's no limitations. And with new genetic tools, so you might have heard of this genetic tool called Crispr Engineering. So Crispr is actually the immune system of bacteria, because like we fight viruses, like measles viruses, bacteria are permanently infected with viruses. So they're called bacteriophages. And these bacteriophages try to kill bacteria. And the bacteria has developed immune systems to fight the viruses which kill them. This immune system is called CRISPR. And over the last years, and probably this year, the Nobel Prize will be given for this, it became super easy to actually take these bacterial genetic tools to cut and paste basically any gene you want in any species. So what I'm telling you now, it's possible to do genetic engineering in your garage, which of course has a lot of issues on, can you create a virus which might wipe us out? Can you change genes in little embryos to make them run faster? So there are lots of little issues which we're actually facing now because of this. But that's the first revolution. We can read genomes and we can actively change them in basically any cell type in any organism we are facing, which we can grow. So 3-4 years ago, this was not possible. The second evolution and revolution was this little experiment for this guy who also won the Nobel Prize for this, where he could make induced stem cells. So the grand paradigm of biology was always we are born out of one cell. So the sperm comes, the egg comes, there's this first cell, and out of the first cell, we develop as a human being. So as a little embryo, which the cells develop in brain and muscle and skin and liver. But this was basically one directional, like time. We start here, and after my talk, time has passed. So biology was one directional. So what he did, and what we can actually do now, and everybody can do now who has the tools in the laboratory, we're actually starting a huge stem cell lab in our institute in Vienna. So what he could do, you take, and I can do it from all of you if you like, take a little drop of blood or a little biopsy of your skin. All you need to do is cut in 4 genes in the cells, and 4 weeks later you have stem cells. So basically what he opened, he basically reverted time in biology. In a 90-year-old person, you can grow a stem cell. A stem cell out of which every cell can be born again. So this paradigm that we are born with one cell and go in other direction can be totally reverted with everybody who sits here. That's the second huge revolution which happened. We can read genome, we can change them, and we can revert the time of biology back to the first cell out of which we came from. And the third revolution, I believe, is this one. We learned how to grow human tissues. So this is the first human brain grown from a stem cell. So this was actually done here in Vienna, right here in Vienna, from my deputy director, Jürgen Knoblich. One day he came to me, showed me on the iPhone, he said, I have to show you this picture some years ago. And I said, this looks like a brain. And he said, yes, it is a brain grown out of a stem cell and tissue culture. So what's possible now, you take a human stem cell or an induced stem cell, I can actually take cells from Markus, grow them back a stem cell and make a little brain of Markus. And so what you need to do is you need to do tissue engineering, bring them to some progenitor cell. And the amazing thing is this cell at some stage actually know what to do. So they have this biological building plan built into themselves. They know they have to become neurons, brain cells. They know to grow in which direction. They know to form synapses. They know how to talk to each other. So we can actually learn from these experiments, from building plans of nature. And this also totally revolutionizes now neurobiology, because we could not experiment on human brains. But now you can actually take from people with autism or schizophrenia or epilepsy or brain cancer, you make stem cells, grow them back, make little brains, and actually study the fundamental mechanisms of disease. And maybe one day come up with new therapies to do this. So this is what we did in my lab now. Can we engineer human blood vessels? And I just show you this. So these are actually the first human engineered blood vessels. So it's not published yet, so that you cannot find it in the published literature. So one of my students managed to make perfect human blood vessels, capillaries with cells inside, cells which line it outside, and a fine membrane around it. So we can even take these human blood vessels, transplant them back into a mouse, and we grow now mice which have human blood vessels. So tissue engineering. Not yet. So we are not doing this, but I know a group in the US is trying to do that, yes. So now I shocked already Francesca, but now I try to de-shock her to show her the reason why we actually do this. Because at the end, you always have to ask yourself the famous question of, what are we doing here? So why are we actually doing all of this? And of course, there's Dostoevsky's Karamazov's stick. One end might not be so nice, but the other end might actually get us somewhere else. So the reason why we do this, and I'm finished very fast. So there are 420 million people with diabetes on this planet already. And you are traveling Polynesia, and this is a huge problem. In Samoa, in many of these countries, people over evolution have been adapted to the environment, to the food, and all of a sudden, Western food comes in, the supermarket comes in, and the adaptation to our environment does not work anymore. That's one of the reasons why diabetes is totally exploding. 420 million with diagnosed diabetes, another 500 million already on this planet who are actually pre-diabetic and just ready to get diagnosed with the disease. So 50 to 80% of them die because of the blood vessels, because diabetes changes the blood vessels. So the blood vessels get very sick. Because they get very sick, oxygen cannot go through, the tissues cannot be nurtured anymore. That's the reason why patients with diabetes cannot heal wounds anymore, why their legs need to be amputated, why they get strokes, why they get blind. In some of the communities, I think on the islands, 10% of the communities are people who are blind because of diabetes. So that's why we do this, because with our tools, we want to figure out how it works. And if we figure out how it works, maybe we can actually do something against it. And so, however crazy this is, what it might look through, but that's the reason why we do this. So we made, we can grow these blood vessels now, and we gave them a diabetic medium. And to our amazement, these blood vessels we grow in tissue culture, human blood vessels, change their structure, like humans with diabetes. So nobody has ever done this before. So now all of a sudden we have a system where we can actually model diabetic changes in blood vessels. and may be able to do something about it. This is human. No, this is all unpublished. Nobody even knows whether we can do this. So you hear it the first time here. And the beauty is, yeah, but this is the next step, exactly the same thing. So can we do something about it? And we can. So on this side, you see that's normal blood vessel. In the middle is diabetic. The green is actually the membranes around the blood vessels. They're all green. They expand like crazy. And then we actually found a drug which works. And they're perfectly normal again. And of course, it's a long, long way from an experiment like this to test it in a human. You know, needs to. We are doing it right now. So in 2 months, I can tell you if it works. So, no, this is human blood vessel we grow in tissue culture in the labs. Exactly. That's why he asked if this can be done in a real situation of diabetes. But so there are many people who work there. So go back to the Marquesas. So this beautiful place there. And one thing we actually developed when we were on the last expedition and talking to people was, we live in this world which we as humans have been changing in terms of the climates are changing. One of the reasons why we actually study viruses is, and why the European Union supports our virus research is, because microclimates in Europe are changing. Meaning there are viruses now coming to Europe, which we have not seen since centuries. And so we better as a population figure out how they work before we're getting surprised by this. So climate change, for instance, allows that certain mosquitoes all of a sudden can live in Europe again, which brings certain diseases. So climate change is not some harmless little thing. Of course, we all know it's not harmless, but in my field, this has direct implications for our health. Because the last thing we want is climate change brings viruses to us, which wipe us out as a population. So in addition, of course, to changes in water level and what this all does to people and their livelihoods, but this is another issue. So we live on this planet with climate change. We have all these beautiful tools learned, changing genes, reading the sequences, the genetic code of organisms, making models where we can possibly figure out disease. And there are these beautiful, unique environments, like on the Marquesas, like on Galapagos. Some places where we can learn so much for this planet. We can learn about diversity in plants, find new plants which maybe help against diseases. Artemisinin, the best drug against malaria, was found in a plant. So there's not. Oh, plants. So this must have been a Freudian little slip on that day. Don't know what was on my head. So here my grand idea, and we already shared it. So what about if we all together actually hook up and build a Darwin station on the Marquesas? At one of the most unique places on the planet, a station which has all the high tech, with all the sequencing, which is totally open to every scientist on the planet, which supports the grandest ideas in marine biology, in diversity on plants, in diversity in humans, in figuring out which plants could be used against disease. Basically provide everybody to go there, learn the unique environments, and learn from this environment the secrets of this planet. And if we have figured out the secrets of the planet, so maybe we can actually gain some years of our life back. And that's the whole idea, and it would be a tremendous opportunity to do this. instead of going, which is of course essential, try to convince the United Nations to do something, but you have been there instead of talking, instead of drinking coffee, us concerned population doing something. And this could be a little contribution to figure this out and actually help protecting our planet. Not just for us, but also for the people living there. Changing ocean levels in Vienna might not be entirely relevant, but for these people, it's the livelihood and the future. So I do believe, and I showed you some of the examples, what's possible now, if you like it or not. But this is our world. This is the world we live in. And the question for us is, can we use these techniques and technologies to the better of mankind, to the better of our little planet, to really make a difference in the world? And I always like this, and I now finish. One of my favorite poets, Italo Calvino, one of my favorite books, Mr. Paloma. So, we're all modeling how the world looks, but the reality and nature are totally different. And that's actually the beauty of nature, because, we can dream many times to believe we have figured it all out, and reality and nature will just slap us in the face and tell us the world works totally different. And so with this, I think I stop and give it back to Markus.
Yeah, I hope you will agree that the plasticity of truth comes only from various disciplines together. Yeah? And language is a magical thing. So speaking about sequencers and samplers and veins and waves, we come to Peter, who today he said to me quite interesting thing that he was the first in Europe, if I'm in the world, to have a computer at home. The home computer. Yes. Andrew has also a quite interesting background: anthropology, social archaeology, marine law, and field recording. So, in a paradox, I would like you start briefly to describe like how it comes, like to come from anthropology to field recording, and what is the sense of it before we come to the most legendary person in sound and digital. Okay.
Of course. Thank you, Boris. And thank you all for having us here. Hi there, Peter. Excellent. Great. That's right. As Boris said, I've in my limited years so far, I've done a little bits of a few different things, and which has included. That's right. I studied social anthropology and archaeology at university, and I did my final project on life at sea, and I lived on board a cargo ship. I have a Greek background in the sea, and I then decided to set up a little record label called Depth Sound Recordings in order to explore that further, to explore both anthropological, ethnographic study of all sorts of people around the world through sound. I have spent some time traveling to interesting places, including Papua New Guinea, where I've done some field recordings and recordings of various tribal musics, including of a crocodile skin cut scarification initiation ceremony in the Sepik river system and deep in the jungle there. And I've manipulated a few of these sounds into various different performances and things. But more recently I've decided to try and combine the sea with this exploration of sound. I had a good friend, Susie Buchan, who was an oceanographer off the coast of Chile, working in the field of whale migrations, and she had done some dipping hydrophone recordings of these blue whales. And Chilean blue whales are actually the only blue whales whose song is fully audible. A lot of whales create sounds that are inaudible to human hearing, but the Chilean blue whale, you can actually hear the whole frequency of their sounds. Soon after encountering these sounds, I thought to pass them on to Peter, who I had recently met. This was a number of years ago, about 2012, and Peter excitedly said that he'd be interested in creating a new composition, and this is the composition that we'll be hearing shortly inside the gallery space. It is composed exclusively of these blue whale sounds, and they're very unusual sounds, actually. Peter will, I'm sure, tell you a little bit more about the sounds and how he's encountered them and used them and manipulated them, but they are not like the humpback whale or other whales that we are maybe more familiar with. They're not like these these humpback whale songs. They're quite different. Peter, how did you find the sounds when you first heard them?
I was going to say it a bit later, shall I?
Sure. Yeah, I mean, absolutely. I mean, I can give
written out. So I've been a bit.
Would you like me to? Why don't I give a little background to Peter? Peter's. I want to give a little bit of Peter's background as well, if I may.
Yeah.
Sure. In about 1960, as Boris mentioned earlier, half a century ago, Peter set up the first electronic music studio, or an electronic music studio, which at the time, it wasn't just any music studio. It was, aside from the sort of the myriad oscillators and tape recorders and things in this studio in London, was the first personal computer in the world, as was alluded to earlier. Over time, a variety of different musicians passed through the studio, and in a couple of years it became the place to go for all sorts of rock and pop musicians, from Pink Floyd to the Beatles. As Peter's interest developed, and as the innovation progressed, as did his ambitions, I think, he decided to start developing synthesizers for the mass market. One of the most famous is the Vcs3 synthesiser, which was a portable synthesizer, used on albums, all sorts of albums, Revolver and all sorts, I'm sure you're aware of. So I think the idea behind this was to allow the exploration of this new field of music, the electronic music, by the masses, by institutions, by schools, by people who could afford to have these in their homes and in their families. So it really shouldn't be underplayed just how important Peter's work and inspiration has been to the history of electronic music. I hesitate to say, but probably the genre as we know it today wouldn't be in existence without Peter's work. So it's been an incredible privilege to work alongside him and collaborate on this piece of music that we will hear very shortly. One last thing before I hand it over to you, Peter, is I think it's important to highlight an environmental reason behind the piece of music here. Actually, the habitat of the blue whale and of all marine creatures is, as I think you'll all know, probably is changing, and blue whales in particular are suffering from things like not only marine pollution and climate change, which is affecting the krill levels of krill, which was their only prey, but also they are struck by ships quite often, and they are affected a lot by ship noise, and which a lot of people don't really realize how noise pollution affects the migration of these the largest creatures to have ever lived on this planet. And with those thoughts, I'll pass it over to Peter and can say a little bit more about the piece and anything else you'd like to say.
Right, thank you, Andrew. Well, thank you, TBA 21, for the chance, really, to present this piece as an ambisonic, which is a surround sound, amazing setup of speakers and things, which I'll talk to you a little bit about. So, we showed this piece, gave this piece in Athens in Documenta a few weeks ago, but that was completely different. That was for 4-channel sound, and I've been working on this production since Easter, which is a multi-channel presentation where the loudspeakers, although there are many of them, shouldn't really appear to be there at all. You shouldn't notice them. So what happens is that I prepare a large number of tracks, and then these, with Professor Myatt at Surrey University, are transformed into an ambisonic, it's called, format. And then these are brought here, and another computer here converts this ambisonic track using Max MSP and its own dedicated computer into the sounds which are then transmitted to the loudspeakers. But anyway, thank you, Andrew, for having introduced me to this amazing sound material. First of all, he gave me a large amount of burbling oceanic sounds, tweets and burbles and squawks, and he said there are some whale sounds here. And first of all, I thought, well, there is occasionally a sound like a sort of hoot, ooh, ooh, something like that. And I thought, well, that must be. I'll keep those. But obscuring them all the time was a sort of terrible repetitive motorbike. And so a lot of my time was taking away this motor. But how could they be recording these whales, I thought? With this motor running, surely they would turn the engine off when they were recording it. But of course, then I sent this first samples to Andrew. I said, here, I found the whale song, the little hoot. And he said, no, the motorboat was the whale song. So the whale song starts with this repetitive, extraordinary sound, rather like if you knock on a door peremptorily. It's like that, pop, 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 or gorilla going like that. And it repeats itself. And then at the end of this sequence of thumpings comes this little cry of joy, as it were. Whoo! But the strange thing is, it's only the males who make this sound. So we don't know what effect such a terrifying or beautiful sound might have on the females at all. Anyway, out of all the hour-long sounds, or whatever it was that Andrew gave me, I decided to just analyze one complete whale sound, take away all the noise, and then use this as a source for a series of computer variations. I only used a computer, not any synthesizer, and make a series of variations on this sound, high, low, stretched out, whatever you like, and make a multi-track version of it. And I tried to express also the sort of dying cries and the birthing sounds of whales. So with luck, we get the whole spectrum. What would be good is if you imagine that you're in a sort of bath when you listen to this, a bath of oceanic sounds, and just sort of relax with these sounds. But occasionally, not looking particularly to where the sounds come from, just get out of a bath and wander around a little bit too. There are 2 rooms with slightly different sounds in each. So you can move from one room to the other, but not running or anything, just gently, and then go back into the bath and relax and until it ends. At the very end, it's half an hour long, this piece. At the very end is the purest whale sequence I could extract. Anyway, I hope you enjoy it. Let's go and hear it.
Thank you, Peter.